Yeah, I'd wait a little longer, but then, so it's just you and Steve, right, so far?
Yeah.
Okay, well, if no one else responds, that will be your group. But let's hope that the others will. Anyway, let me know one way or the other what's going on. I'm keeping a record of all the groups.
All right, no problem.
So that's one of the things I wanted to announce for everyone is I know that some of you have started organizing your groups. I don't know if you all have, but I want to encourage you to do that. And let me know if you have any problems. Just send me an email if you have any problems organizing your group. So that's one point. Another point is I've assigned a worksheet. It shows up in Canvas as an assignment on cell division. That's what we're going to talk about today. And on Wednesday, the 13th, we will have a quiz.
When?
Wednesday.
Oh, God. Okay, the worksheet that you are putting out today, that's due tomorrow, Yolanda?
The worksheet is, I think it's due tomorrow.
And then a quiz Wednesday.
Quiz Wednesday.
And it's on the same thing, the worksheet?
Yeah, it's a little more than just the worksheet. So the quiz on Wednesday will cover cystic fibrosis. And for that, focus on the pancreas and gallbladder.
Oh,
That,
okay.
The several page document that we covered on the gallbladder, what its role is, and the pancreas. It will also cover the quiz on Wednesday, coronavirus and vaccines. So make sure you know how the coronavirus infects, what the mechanism is, and the different types of vaccines that are available. And then there'll also be a question on membranes and transport, which we went over last time. I'll not put anything about cell division, but make sure you understand about fluidity, what makes the membrane more fluid or less fluid. And that's important because the fluidity determines what can pass through across the membrane. Okay, and then test 2 will be on October 27th. So write that down. October 27th is a Wednesday. There'll be no class that day because the entire class period will be devoted to the test. And that test will cover everything from mutations and including CRISPR, everything from there through Mendelian genetics. So we haven't done cell division yet. We do that today. We do Mendelian genetics next time. And then there's going to be a review. The previous class period before the test on October 25. So I've sent you an assignment asking you to send me questions. And the due date for that would be the Saturday before. So the Saturday would be October 23rd. Send me your questions and we'll spend the entire period on Monday, the 25th, discussing your issues. And then the next class period will not be a class period, but Wednesday, the 27th will be test 2. So any questions about what's coming up? All right, there's quite a lot I want to cover today. And so for that, I'm going to share my screen with you. Cell division, mitosis and meiosis. So in order for organisms to do the basic things of life, like grow and develop, reproduce, cells have to divide. And these two processes, mitosis and meiosis, ensure that genes are transmitted faithfully from one generation to the next, on the one hand, and on the other, an emphasis is going to be on how variation is introduced. But first, before talking about meiosis and mitosis, let's talk about cell division in prokaryotes, where mitosis and meiosis do not occur. And that's because prokaryotes have no nucleus. They have an area where the DNA is found called the nucleoid, but that is not membrane bound. So it's not a nucleus, and it doesn't have mitosis and meiosis. So in this figure, you can see the steps to binary fission. That's what it's called when prokaryotic cells divide. It's called binary fission. So number one, the cells grow, and then DNA replicates. So you start with one strand of DNA, and you end up with 2 strands. And that's basically it. The cytoplasm divides, the cleavage furrow occurs in the center, new cell wall is made, the 2 cells separate, and over on the left here, I've circled the word identical because what you end up with in binary fission are 2 cells that are identical to the progenitor. So the cell over here at number 1 is copied precisely for number 6. So keep that in mind, that prokaryotes divide by binary fission. Eukaryotes do have nuclei, and it's within the nuclei that either mitosis or meiosis occurs. So let's first consider mitosis. What are the consequences of this? Well, you start with one cell. It can be either haploid or diploid. It duplicates its DNA and divides. And the result is 2 cells identical to their progenitor. So just like binary fission, the result of mitosis, even though it's much more complicated, the end result is the same. You get 2 daughter cells identical to the parental cell. And no genetic variation is introduced during mitosis, except occasionally there might be a mutation, but that's not systematic. Now this slide has a lot on it. I'm going to go through it slowly, but these phases of mitosis you need to know. So know what the name of each phase is and know what happens in each phase. So first look at interphase. Cells don't look like they're doing anything. They're just sitting there microscopically, but there is activity, and I'll go through the steps of the activity that go on during interphase. But once interphase is over, prophase starts. And here the nucleolus goes away, and the chromatin condenses into microscopically visible chromosomes, these little worm-like structures. And then prometaphase, the nuclear envelope breaks down, and the sister chromatids, in other words, each chromosome is now copied, and the copy is called a chromatid. They're joined by centromeres. And you see a drawing of that here at the bottom, this orange structure, 2 chromatids, which constitute 1 chromosome, joined by a centromere. The next phase is metaphase, where all the chromosomes are now highly condensed, easily seen in a microscope, and they line up in the center of the cell in one plane. The next step is called anaphase, and that's where the chromosomes separate. Half of them go to one side of the cell, and the other half go to the other side. And finally, we have the last stage, telophase, where the chromosomes arrive at the so-called poles of the cell, and these spindle fibers, these pink things, disappear, and we're ready for the last step, which is called cytokinesis, and I'll talk about that in a bit. But before doing that, I'd like to elaborate a bit on up in the upper right, the anatomy of the nucleus in interphase. So here you see this large purple thing is a nucleus. The dark purple is the nucleolus, and these little strings are called chromatin. So what is chromatin? It's really chromosomes, but they're very relaxed and very long. And in cell division, the chromosomes condense so that you can actually see them in a microscope. But chromatin is the same thing as relaxed chromosomes. The nucleolus, this dark purple thing inside the nucleus, what is the function of it? Well, it makes ribosomes. And the ribosomes, after they're made, are sent out of the nucleus into the endoplasmic reticulum, which you see here, this gray area. And it's in the endoplasmic reticulum that the ribosomes and the transfer RNA and the messenger RNA all get together to produce proteins. And this is a process that we went over previously, so you know how this works. So that's what I want to say about the structure of the nucleus, the basic structure, the nucleus, the nucleolus, and the endoplasmic reticulum. Any questions about these structures and what their functions are? Structure and function should always be considered together. All right, I want to go now and focus on interphase and describe what actually happens in interphase. So remember, we go back, interphase is just this first point where mitosis starts. And to look at the cell under a microscope, it looks like nothing is happening. But in fact, it's divided into 3 phases. G1 is the growth phase. During G1, the cell is growing in size, and it's synthesizing enzymes and the nutrients that are needed for cell division. The S phase is synthesis. This is when DNA is replicated. So recall the 6 steps of DNA replication. We've gone over that several times. And remember that copying errors can occur when DNA replicates. Those are mutations. So in the next phase, G2, the cell checks this replicated DNA for copying errors. And if it finds them, it tries to correct them, sometimes successfully, sometimes not. So mutations may or may not survive the transition into mitosis. And the last phase then is entry into prophase, into the so-called M phase, mitotic phase, and that is here. That this transition from interphase to prophase is what's called the M phase in this diagram. So after G2, or after mitosis occurs, cytokinesis occurs, and I'll discuss that now. In animal cells, the 2 daughter cells is pinch apart. There's a constriction between the 2 cells. What's called a contractile ring is made, and that pinches off to make 2 cells. So this is cytokinesis in animal cells which do not have a wall. In plants, it's different because they do have a wall. So what happens is that after the chromosomes have gone to the opposite sides, the cell starts to make cell wall material and assembles the material into what's called a cell plate. And that fuses then with the walls of the cell to make 2 daughter cells. This is cytokinesis in plants. The difference being that the wall has to be accommodated.
Real quick, what are the vesicles and cell plates made out of typically? Like, or like, how do they begin to form in this process, I guess?
Alex?
Oh, so Anthony Emilo?
Oh, Anthony Emilo, okay. Okay. What are the materials that make up the cell plate?
Yeah, is it just like proteins or?
Mostly carbohydrates.
Oh, okay.
Remember, the cell wall in plants is made of cellulose, primarily cellulose. And so to make new cell wall, sugars have to be polymerized into long chains. And that's what goes into the cell plate, which then fuses with the wall to form 2 daughter cells.
Okay. And if this was a plant, would it be like creating more cells just because it's getting larger essentially, or?
Yes, mitosis in plants is when the plant grows. So
Okay.
some plants like corn, for example, on a hot summer day, if you go into a cornfield and you listen, you can hear creaking sounds, and that's actually the sound of the corn plant growing.
Oh.
Wait, so the cell plate does not disappear, it just becomes part of the wall?
Yeah, cell plate fuses and becomes part of the wall.
Okay, just making sure.
Okay, anything else about mitosis?
Professor,
Yeah,
maybe, I mean, it has to do with mitosis, but it's kind of a weird question, I think. It's the growth, like, it's just a growth tissue, right? It just multiplies and it grows.
yes.
Does that mean there is a like mistake in mitosis when a tumor forms? Like, is that what it is?
Yeah, when cancer occurs, it's actually cancer is the result of several mutations. And these can be the result of any number of things, but cell division could be one of them. Mistakes could be made in DNA replication and mitosis and initiate a cancer.
Okay.
Another question. Now, so this is cytokinesis. This is what happens when a plant is multiplying, but is there cases where it's actually being reduced? Is it the opposite also possible where they like are almost destroying themselves or something?
Yes, there is a process known as apoptosis, which in fact is just what you said. Cells destroy themselves. This is a necessary part of development. It's something that we're not going to go into in this course.
Okay.
But that is a very good point to be made. And that is that we are putting all this emphasis on growth and cell division. And that, of course, is important, but equally important is that cells commit suicide. They destroy themselves when it's necessary.
Okay.
So an example of that would be in a fetus when fingers form. First, there's just one big tissue, but then the cells between the fingers have to die.
Really? That's I didn't know that's how those were formed at all.
Yeah, if not, you'd have mittens instead of fingers.
Yeah.
Okay, but that's apoptosis, and it's something that's more detailed than we're going to deal with in this course, but it's a good thing to point out. And if you want to know more about it, you can Google it and find out.
Gotcha. Thank you.
All right. Now, before we get to meiosis, we're going to go through in detail the steps in meiosis. But before doing that, I want to consider the human life cycle and the fact that we humans, like all other eukaryotes, can exist as either haploid or diploid. And I want to consider the difference between males and females in the haploid stage. So let's look first on the left here. Females are born with many thousands of egg cells. Now, egg cells are the products of meiosis. Many of these die, but at the time of birth, baby girls have thousands of viable egg cells. And then later on in their life, at puberty, the menstrual cycles start, and one egg is shed for every cycle. So there are about 500 total eggs over the course of about 42 years. One is shed each menstrual cycle. And eggs can actually be produced after birth, but it's sort of a minor source of eggs in most women. And the reproductive phase for women ends with menopause. So there's a distinct end of the reproductive phase of a female. And you can see the cycle here, the haploid cells, the egg, the ovum is haploid, the sperm is haploid. And so it's only during the sperm and egg phase that we exist in a haploid condition. Then when fertilization occurs, we have a diploid, and from that is one cell after fertilization that is diploid. And as it grows and develops, we achieve the development of our full bodies. And all the cells in our bodies are diploid, except for the reproductive cells. Now, contrasting haploid in human males, they're born without sperm, no sperm at all until puberty. They begin making sperm. It takes 2 to 3 months for a male to make a mature sperm cell, but they make millions per day. Even though it takes a long time, the development is staggered so that there are millions of sperm made mature every day. And males, in contrast to females, don't have a distinct end to their reproductive phase. They can go on making sperm until they're very old. In some males, reproductive phase ends. In other males, it doesn't. Okay, any questions about that? I want to just contrast the difference between the haploid phase in males and females in humans.
So you say that the females stop making eggs or can't produce children when they hit menopause, but the male sperm continues to be able to produce well over old age.
Yeah, you probably all know of examples of a fairly old man, say 70 or 80 years old, married to a woman who has not entered menopause yet, and they have a baby. And that's because men can keep making sperm indefinitely, although the rate of production of sperm decreases with age, and the chances of mutations increase with age, but sperm production continues.
And with that, like they have where they say after a certain age, if a woman has a child, something happens to, other things can happen, deformity or whatever to the child. But with the male sperm, it doesn't have that effect after he gets a certain age, that it can produce deformities in children past a certain age.
Well, the chances of mutations in sperm, the probabilities increase with age. Now, that's also true in females, that the older they get, the eggs that they produce are more likely to be defective. So that's why usually a woman who becomes pregnant near the end of her reproductive phase usually has her fetus checked out for obvious deficiencies, because the probability of them is higher in an older woman than in a younger one.
And that's because you said the egg is getting ready to decrease or getting ready to die. That would be the reason why it would be
No,
that.
it's not because it's getting ready to die. It's because as eggs are maintained, remember, a female is born with enough eggs to last throughout her reproductive phase, even though new eggs can be produced. So the older the woman gets, the more likely it is that there's a defect, a genetic defect in the egg cell.
I have a quick question.
Yeah, your name?
Yusuf Faraj.
Faraj, okay.
When it says n equals 23, does it just mean the pairs of chromosomes?
Yeah,
Oh, okay. Just making sure.
yeah. So the haploid chromosome number is 23, diploid is 46. You see down here where it says diploid zygote, 2n equals 46.
What's the difference between that and the multicellular diploid?
Between that and what?
The multicellular diploid. Because it also says 2n equals 46.
At the bottom, which is multicellular.
Yeah, so all the cells, after the zygote is formed, so after the sperm fertilizes the egg, you have this diploid zygote. From then on, every cell in the body is diploid, 46 chromosomes.
Oh, okay. Thank you.
So we exist almost our entire lives as diploid organisms. And it's only in our gonads, our reproductive cells, that meiosis occurs and you get these haploid gametes. But they never develop into real organisms because fertilization occurs. And that brings up another point I want to make. I've just been emphasizing that we humans spend most of our lives as diploids. And so we're diploid for literally decades. We're haploid for as little as a few hours or a few days, depending on when the egg or sperm is made and when it's used, when it might be used. And in contrast to that, there are other types of organisms that are just the opposite. They spend most of their lives as haploids and are diploid for a very short period. And examples of that are many fungi, which will form what's called a zygospore. And that's diploid, but it immediately goes into meiosis and makes haploid spores and haploid other cells, and it can exist for months, years, decades, even centuries. It can exist in this haploid state. And in fact, some fungi are not known to have a diploid phase at all. So just remember that humans and many other eukaryotes are diploid most of their lives, but there are eukaryotes that are haploid most of their lives.
Professor,
Uh-huh.
I have a question. This is Gabriela Galvez. Which cells are haploid? Which cells?
Which cells are haploid?
Are haploid.
The cells that are haploid are the eggs and the sperm.
Okay.
So, and we're going to go into the details of how you make a haploid cell, because that is in fact meiosis. So it says here on the left, meiosis gives rise to haploid eggs in females and haploid sperm in males. And then when a sperm fertilizes an egg, now you have a zygote which develops into a fetus and is born and develops into a full-blown human.
Okay, so separately, they're the haploids, and then when they join together to fertilize, they become diploids, zygotes.
Yes.
Okay.
Okay, now we're going to go through the steps in meiosis. How do you get a haploid cell from a diploid cell? So the consequences, the cell starts out as diploid and it ends up haploid. The chromosome number is reduced by half. 2n goes to 1n. And the other important part of this is that genetic variation is created. And I'm going to emphasize that as we go through. Where does this genetic variation come from? Because this is not random mutations. This is systematic introduction of variation into the cell. So let's first look at meiosis 1. There are two meiotic divisions. They're called meiosis 1 and meiosis 2. So if we go to the left here and we have interphase, this is the end of interphase. And you remember in interphase, all the DNA is copied. The cell grows. The DNA is checked for errors and so on. But then you go into prophase. And so this is showing 4 chromosomes, two long ones and 2 short ones. And each chromosome is actually 2, because remember the DNA replicated here in interphase. So now each chromosome is actually double. And a doubled chromosome is called 2 chromatids. So 4 chromosomes equals 8 chromatids in this cell. That's in prophase. And genetic variation can occur here due to crossing over, which I'll describe in a little bit. But the next phase then is metaphase 1, where the chromosomes line up in the center of the cell. And again, genetic variation can occur at this, in fact, it does occur at this phase. It's called independent assortment. But we still have 8 chromatids per cell. Now we have anaphase, where they go to opposite poles. And so now that this is haploid, we've gone from diploid to haploid. If you're looking at chromosomes, so we started out with 4 chromosomes per cell. We end up with 2 chromosomes per cell, but each one is doubled, so it's 4 chromatids. And then the cell divides so that we now have 2 haploid daughter cells at the end of the first meiotic division, meiosis 1. So it will become a little clearer, I think, as we go on. And at this point, I'd like to go back and emphasize the sources of genetic variation. One source of genetic variation is the fertilization step. Fertilization resulting from sperm and egg joining. So if we go back to here, if an egg is shed during the menstrual cycle and it encounters millions of sperm, each of these sperm is genetically unique. So I'll show you in a minute why that's true. Each sperm is genetically unique. And so it depends on which sperm actually does the fertilization as to what the genetic constitution of the zygote will be. So this is a source of genetic variation. Fertilization itself is a random choice of sperm. Each sperm is genetically unique because it was created by meiosis. Now within meiosis, there are 2 other sources of variation. One is called crossing over, and the other is called independent assortment. So let's first look at the right here, crossing over. And this occurs in meiosis 1 in prophase. So that homologous chromosomes come together. And in this case, for illustration, you see that we have a blue chromosome and a red chromosome. Then, because they formed a tetrad and they're in intimate contact with each other, they exchange parts. And these points of exchange are called chiasma, but that's where crossing over occurs. And so what you end up with metaphase, when the chromosomes separate, you now have some chromosomes that are composed, they're chimeras. They're composed of parts of both chromosomes. So we have here one chromosome has a blue chromatid, and another chromatid, its homologue is now hybrid. It's picked up two red pieces, so it's partly blue and partly red. And correspondingly, in the other cell, one chromosome is unchanged, it's wild type, it's all red, whereas the other chromatid, it's a homologous chromosome, is recombinant. That is, it's partly blue and partly red. So if you look at the gametes then, the gametes would be either eggs or sperm. Some gametes have just one blue chromosome, and the other one is recombinant. And another gamete can also be recombinant, but the opposite. This is red, blue, blue, red. This is blue, red, red, blue. And then there is an unchanged, a cell with an unchanged chromosome. So it's these recombinant chromosomes that are the source of variation in crossing over. And crossing over always occurs whenever meiosis occurs. And the other source of genetic variation is called independent assortment. And this can happen in either or both of the meiotic divisions. So what does independent assortment mean? Well, you see here, there are two possibilities for this cell. So you can have a blue long, a red long, a blue short, and a red short in this configuration, so that when the chromosomes line up in metaphase, you have long blue, short blue, long red, short red, and the combinations end up just like possibility one. In other words, one egg or sperm is long, short blue, long, short blue, long, short red, long, short red. However, if when they line up, they switch positions, you now have blue on one side, a long blue on one side, red, long red on the other, and they switch. So the short red is on one side, short blue is on the other. Now, if you follow this through, when the chromosomes separate, you now have combinations 3 and 4, where instead of blue, blue, you have blue, red, 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 blue, red, blue. So between these two processes, independent assortment and crossing over, you have two sources of genetic variation in meiosis. Now I'd like to zoom in on how crossing over actually occurs. It's an exchange of parts of non-sister chromatids. So these are two chromosomes, a red one and a blue one, and each one has copied its DNA. So they're called two chromatids, sister chromatids. But blue and red are non-sister chromatids. So this is a duplicated maternal chromosome and a duplicated paternal chromosome, and they form a tetrad. Tetrad means 4. So there are 4 chromatids in this tetrad. And this occurs with every homologous set of chromosomes. So in a human, it would be 23 of these form, one for each pair of chromosomes. And while they're in this intimate association, they can exchange parts, so that you end up here with an unchanged homologue and a recombinant blue, red, blue, and another recombinant, red, blue, red, and then an unchanged chromatid. So these recombinant chromosomes are now unique. They're different from either parent. And now in the second meiotic division is meiosis 2. Remember, we ended up, go back to here, we ended up at the end of meiosis 1, we have these 2 haploid cells. But the chromosomes in each of these haploid cells are actually double. And now here are those 2 cells that we ended meiosis 1 with. They go on to divide again, and that's called meiosis 2. So you have telophase 1, the end of the first meiotic division, then prophase 2, which is the beginning of meiosis 2, and then at metaphase 2, the chromosomes line up, and half of them go to one pole and half go to the other. And the important thing about this metaphase 2 is that which pole a chromosome goes to is entirely random. There's no prediction, no way to predict which side of the cell a particular chromosome will go on. So, and then the cell, anaphase occurs a second time, and the chromosomes go to the opposite poles of the cell. Then the 2 cells divide, so now you have 4 cells, and you end up with wild type and recombinant chromosomes in each of these 4 cells. So each of them, the thing to remember about the end of meiosis, the end of meiosis 2, is that each of the 4 products is genetically unique. So remember, I emphasized in mitosis that you end up with cells that are identical to the parent. Here, you end up with cells that are entirely different from the parent, genetically different. All the genes are the same, but the combinations of genes are all different. So if these were sperm cells, each sperm cell is different from the parent. If these were egg cells, each egg is different from the mother. So independent assortment occurs at metaphase. At metaphase 1, the chromosomes go randomly to the poles. At metaphase 2, they do it again. The decision which chromosome goes to which side of the cell, it's an entirely unpredictable and random process. And so this is a very important source of genetic variation. Now this table compares the 2 meiotic divisions, meiosis 1 and meiosis 2. So meiosis 1 starts with a diploid cell and ends as haploid. Meiosis 2 starts as haploid and ends with haploid. And that's because of the second meiotic division, which is called the equational division. The first one is called the reductive division, where you go from diploid to haploid. Equational, you go from 2 haploid cells to 4 haploid cells. In meiosis 1, the homologous pairs of chromosomes separate. Underline, the chromosomes separate. In meiosis 2, it's the chromatids that separate. Remember, a chromatid is a product of the copying of the DNA. In meiosis 1, crossing over can occur. Meiosis 2, there is no crossing over. But in meiosis 1, recombination occurs because of crossing over. Meiosis 2, it does not. But random assortment can occur in either meiosis 1 or meiosis 2, or actually occurs in both. What about the sex chromosomes? Remember, females are XX, males are XY. So when a female undergoes meiosis 1, she makes 2 X's, and they divide in meiosis 2, so you end up with 4 products of meiosis. Each one is an X. In the case of a male has either is XY and undergoes meiosis 1. One cell is X, the other cell is Y. When these divide in meiosis 2, you now have 2 X's and 2 Y's. Now, when fertilization occurs, the egg is always going to be XX. If the sperm carries an X, it's going to be a girl. She will be XX. On the other hand, if the egg is an X and the male contributes a Y, the zygote will be XY and the child will be a son. So that's how we end up with 50% roughly of our population being girls and the other half being boys. So this chart is to try to summarize what we've been talking about, comparing mitosis on the left with meiosis on the right. So the goal of mitosis is the exact reproduction of the parent. In contrast, the goal of meiosis is to avoid exact reproduction and get variation with each cell division, genetic variation. So in mitosis, the chromosomes replicate and divide, and you get 2 daughter cells, each is identical to the parental cell. In the case of meiosis, you have crossing over occurs, you have random assortment occurring, and you end up with daughter cells that separate in anaphase, and the daughter cells are unique. You end up meiosis, at the end of meiosis 2, 4 unique haploid cells, none of which is identical to the progenitor. So these 4 cells, these haploid cells, are each genetically different from this progenitor cell where it started. Any questions about that? You may have to sit and think about this to sort it out in your mind how meiosis actually works.
So, Professor, can we say that 2 cells results on mitosis and 4 cells result on meiosis?
Say that again. 2 cells for mitosis and 4 cells for meiosis. That's correct. Yeah,
That's correct. So mitosis is 2 cells, right?
right. You start with 1 and end up with 2. In meiosis, you start with 1 and end up with 4. But
Okay,
remember,
thank you.
in mitosis, the 2 cells you end up with are diploid. In meiosis, the 4 cells you end up with are all haploid.
Okay.
Professor,
Uh-huh.
this is Alex.
Okay.
I was gonna ask, what exactly is quiz 3 gonna cover?
You mean quiz 6?
Well, I'm sorry, quiz 6, yeah.
Yeah, quiz 6 will not include any of this.
So
Yeah.
what will quiz 6 like cover then?
But, okay, what it will cover is cystic fibrosis, and you'll want to look especially at the functions of the pancreas and the gallbladder. It's going to cover membranes and look especially at what causes membrane fluidity to change, the causes of changes in membrane fluidity. Going to cover a couple of questions about coronavirus and vaccines. So know what the types of vaccines are that are available in the US and how coronavirus infects a human cell. So there's only 5 questions on quiz 6, and those are the topics that it covers. You get 2 attempts.
Okay, thank you, Professor.
Okay, why is meiosis important in evolution? Remember, evolution occurs continuously on this planet. As the environment changes, living things have to change to adapt to changes in the environment. So meiosis is an endless source of variation upon which natural selection operates to sort out the fitness. So what meiosis does is give natural selection choices. Because variation is constantly occurring, if one variant is better suited than another, that's the one that survives. In meiosis, variation arises from crossing over, that's recombination, and it arises from random assortment in both meiosis 1 and meiosis 2. Chromosomes randomly assort when the cell divides. So why is meiosis 2 necessary? You could say, all right, at the end of meiosis 1, the cell is haploid. Why do you need another meiotic division? And that's because that after meiosis 1, you have a haploid number of chromosomes, but each chromosome is doubled. It's now actually 2, each chromosome is 2 chromatids. And if we didn't have meiosis 2, those 2 chromatids would then double the chromosome number, and it would be lethal for the species. So the equational division, that is meiosis 2, ensures that the organism's ploidy level, that is the number of chromosomes, remains stably diploid. So that's, this is something that you should remember and think about, that meiosis is really essential for evolution to keep up with environmental changes. Now, this is a summary of mitosis 1 and meiosis, or meiosis and mitosis. Not going to go through these. This is, you can consider it a study guide, things that I think are important that you should know. And I'd like to make one other point before finishing this, and that is about human karyotypes. We've gone over karyotypes before. Haploid, 22 autosomes, 1 sex chromosome. A diploid is 44 autosomes and 2 sex chromosomes. This, which one is male, which one is female? Well, females have 2 X's. But then we have abnormal karyotypes. Can you spot the abnormal karyotype on each of these?
Yeah. Like number 5.
Well, I'll point it out. In the case of, on the left, if you look at chromosome 21, what do you see? All the other chromosomes, there are 2 of each. In the case of 21, there are 3. So that's called trisomy 21, and that's the cause of Down syndrome. On the right, you have a female karyotype, and I'll point out the issue here. It's with the X. There's only 1 X, and there should be 2. And that is an abnormality that's called the Turner syndrome. And here you see a little bit more about the effects of these chromosomal aberrations. On the left is Down syndrome, where you have 3 copies of chromosome 21, and this can occur in either a male or a female. It's caused by non-disjunction, which I'll show you in a minute. And there are a lot of different symptoms. All of you have seen people with Down syndrome, and you can see that they have these symptoms. In the case of the Turner syndrome, 1 of the X chromosomes is missing. So this can only happen in females. And the result of it is that their development is slow. They never go into puberty. Therefore, they're infertile. And their life expectancy is fairly short. Now, these are 2 of the trisomies that are viable. In other words, people with chromosome 21 or people missing an X. They're born and they live relatively normally for some period of time. But any chromosome can undergo non-disjunction and end up as a trisomy. So most trisomies, in other words, most chromosomes, if there are 3 copies, they die during pregnancy and abort. A trisomy 18, well, all of these trisomies live fairly short times and they die. So it's only 21 and X that can grow to adulthood. So any questions about Down syndrome or Turner syndrome? So what causes 3 copies of a chromosome to end up in the same cell? It's called non-disjunction. It's the cause of trisomy. So on the left, when a cell divides, so this is a cell undergoing either mitosis or meiosis, you have a blue chromosome, a green chromosome, and a red one. And when in anaphase, when the chromosomes separate, proper disjunction, blue goes to one side, the other blue goes to the other side, one green to one side, another green to another side. But in the case of red, they both go to the same side. And so then when this egg is fertilized, it gets another copy of red, and that means 3, that's too many, and so you get either death or a symptom. Okay. Any questions about any of this? We're going to have a question and answer period later, and you can think about the complexity of meiosis and also mitosis, and whatever questions you have, send them to me. I'd like to stop, and in a little bit of time that's left,
Excuse me, I have a question.
Yeah,
I'm Wasan Rafu.
okay.
And my question is that process of the mitosis and meiosis is just for reproduction cells only?
Yes, yeah, for body cells, mitosis occurs.
Oh, okay.
But anytime that reproductive cells are made, it's meiosis. All right, I want to give you an example of some real research that illustrates a point about cell division. All right, this is I call it the case of the Dynein mutant, and I'll tell you what that is. But it's an illustration of experimental science in action. So this is work that was done years ago when I was at Cornell by one of my graduate students, whose name was Satoshi Inoue. He did this for his PhD thesis. So what's the relationship of this work done long ago to Bio 130? Well, it involves mitosis and it involves mutation. And those are both critical topics in this course. And it also involves data analysis and interpretation. So the question that Satoshi, you can see a picture of him here. His question was, what is the role of a motor protein, in particular dynein, in mitosis? And this is a paper that was published, here he is again, in 1998. So this is work that was done a long time ago. The experimental system that he was using involved a fungus called Fusarium solani. This fungus grows like a mold. It is a mold. And if you look at it under the microscope, you can see that it's composed of these long threads and these structures called spores. Here you see more pictures of spores. And this fungus is actually a pathogen of plants. So here you see it on soybean plants. These are healthy plants. And here, this is what Fusarium can do to a soybean plant: stunt its growth and actually kill it. So the subject of his investigation was dynein. And what is dynein? It's a family of what are called motor proteins. Motor because they move things in the cell. And in this case, we're going to talk about chromosome movement in mitosis. So these dynein molecules walk along microtubules within cells. They use ATP and they move things from one place to another. So here you see a microtubule. You can see a dynein molecule. It can use ATP to walk along the microtubule, and as it goes, as it moves, it carries a chromosome with it. Chromosome is the cargo. And this illustrates that. This on the left is a cell at metaphase, mitotic metaphase. You can see this, the green are the spindle fibers. The blue objects in the center are the chromosomes, and they have to go to the poles. Well, they don't just do that automatically. Some force has to do it. And one of the main forces is dynein. Wherever I've circled this little object, that's a dynein molecule pulling a chromosome toward the pole of the cell. And so Satoshi's question was, what if you take these dynein motors away? What would the cell do? So remember, we're talking now about steps in mitosis. And so these are all the steps, and we're talking in particular about anaphase here. If you don't have a Dynein motor, would this process occur normally? What would happen if one of the motors is missing? The strategy to address that question was to disrupt the gene, to make a mutant. And we didn't use CRISPR. It was decades before CRISPR was available, but you remember the essence is to make a guide to target a cell, or to target a sequence within the genome. So we did that, Satoshi did that, but in a much more crude and laborious way, and that he took the gene, the wild type Dynein gene, and disrupted it. So here you see it's in red, and he disrupted it with a sequence that's here in green, and this sequence just happens to encode resistance to a drug called hygromycin B. And so with this construct that he made, the homologous sequences target the Dynein gene, and he put this into a plasmid, which is a circle of DNA, mixed the plasmid with fungal cells after their walls have been taken off. If you take the walls off of cells, they take up objects readily. So the cells take up the plasmid, and then the cells are spread on an agar medium containing a selective drug. And so this was the whole idea. Satoshi set it all up, and he did it. And he came to me a few months later and said he'd like to have lunch. And so over lunch, we talked about his project. And it turned out that he wasn't getting any mutants. So we thought about that. And I said, well, it seems as though there are two possibilities here. Either your mutation is lethal. In other words, when you make that mutation, the cell dies. And if that's the case, you don't have a project. You have to start out on something else. The other possibility is that the mutant cells are sick. And because they're sick, they grow very slowly. And you're not seeing them because they're being overgrown by cells that are not mutant, that are growing fast. So instead of analyzing his experiment after a week, I said, wait a month, throw away all the fast growing cells and wait a month and see what's growing after a month. And when he did that, he got what you see here. He plated his cells on either medium with no selective drug or with a drug. And you can see that the wild type unmanipulated fungus grows just fine without a drug, can't grow in the presence of a drug. And there are control transformants, that is, that they're not targeted to the Dynein gene, but then the Dynein mutant, you can see, is this poor, pathetic little thing that can hardly grow, hardly grow at all. And it grows equally well with or without the drug because it has the resistance gene. So this is what we could see macroscopically, just looking at plates. Under a microscope, it was quite dramatic. So here you have wild type and mutant. You can see the cell, the morphology of the cells is very different. If you look at the nuclei, in the case of wild type, nuclei are nice, evenly spaced, whereas they're all messed up in the case of the mutant. The spores are all misshapen. And if you look at the movement, the actual movement of nuclei, in wild type, they move very fast from the center of the cell to the tip of the cell. So they move very fast in the controls, and in a wild type, Dynein mutant nuclei couldn't move at all. So this was quite a dramatic result. And the conclusion was that the Dynein motor protein is required for normal mitotic cell division in order for chromosomes to move to the poles of the cell. Now, they can do it using other means, but without Dynein, cell division is very slow, and therefore growth is very slow. Now, again, what's the relevance to Bio 130? Well, it's mitosis, and it's an aspect of mitosis that that is dependent on a particular protein. And if you take that protein away, that was illustrated by targeted mutation, targeted gene inactivation. And this is an illustration of how science goes in the real world. I think that's, yeah. So that's all of that slide set. It's for your entertainment. If you're entertained by a real world example of science, and it goes along with mitosis, which is cell division. So our time is up. Any questions before we go?